Hello friends, welcome to my YouTube channel Knowledge of Biology with Anjali Bute. So today I am discuss with you second stage of the genetic recombination. It's a conjugation process. What is the conjugation process? Okay, so look at that. Last video I am discuss with you what is the transformation and this video is conjugation. Second stage, what is the conjugation? So firstly understand conjugation in this process are obtained by two cells are carried to be physical contact between the each other. Okay, just like. Okay, so this is the F factor and these are present by their genome. For example, so these are two cells are created by differentiation between each other. For example, male and female. So it's a F plus cells. F plus cells means they are donor cells. Okay, they are donated by their genome into the F minus cells. Just like, and then converted into F plus, and then they are converted by F minus. Firstly they are F minus, then they are also converted to be F plus cell because donor cell are give by their genetic material into the recipient cells. They are carried to be physical contact with each other, help of the sex pilus. This portion to be carried between the combination of each other is called as to be sex pilus. Okay, and it's F factor of the donor cells. Okay, just types of the conjugation are carried to be cells. They are genetic recombination. Why genetic recombination are involved to be different cells? Because some factors are present by one strain and some factor are not present by second strain. Okay, so they are carried to be growth on the nutrient agar plates. So that timing this strain are not followed by their growth. Okay, because some ingredient are absent for this strain. Okay, so that timing we are combined to be two strain between the each other and they are complete by their strain. Okay, then they are carried to be their growth on the plate. So we are easily isolated by this colonies. Okay, this bacteria, this strain. Okay, so take by screenshot for this guys. So next step I am consider of you. Okay. So I am consider of few ingredients. Okay. So for example, it's a tube. This tube undergoes are present by one bacterial strain. Commonly we are used by E. coli because E. coli are having to be there different types, different strains. Okay. So we consider to be it's a E. coli type first. Okay. And it's a second tube of the present by second strain E. coli type 2. Both are differently carried by growth onto the nutrient agar plate. Okay. So we observe by into the different strain for the nutrient agar plate. They are not followed by growth. No growth. No growth. Why no growth? What is the reason of the no? Why no growth? Because they are absent by sub ingredients. Ingredients such as, for example, E. coli type first strain have to be biotin plus methionine plus, but leucine are negative and threonine also negative, means they are having to be two strain positive, biotin and methionine, but leucine and threonine are not present for this strain. So second of the type 2 have to be biotin absent, methionine also absent, leucine positive, and threonine also positive. Means two strain are present by to be E. coli type first, and two strain are present by different strain, different their ingredients into the type second. Okay, so they are individually not carried by growth to the another plates. Okay, so they are carried by combined to be each other. Okay, so that timing we are combined to be each other, two strain are combined to be each other. Okay, after some time this strain we are followed by growth onto the nutrient agar plates. So we observe to be just like a colonies growth onto the E. coli bacteria. Means they are combined to be each other. So we are having to be complete ingredients such as biotin plus, methionine plus, leucine plus, and threonine also plus. This is a complete ingredients, complete factor are present to be combined strain. For example, E. coli type first and E. coli type 2 between the each other, so they are followed by growth. Okay, they are followed by growth. So this is the physical contact between the conjugation of recombination process, which steps obtained by them. So I am consider of you, so take by screenshot guys. So I am consider of you next step. Okay, so in this process of the conjugation are also discovered by David scientists. They are used by one of the tube, which is then David experiment. David scientists used by as a David's tube, just like it's a tube cotton centered. Okay, so it is a David tubes. David tubes means they are considered to be two strain, two different strain between the one tube. Okay, one side are present by one strain and second side present by second strain. They are have to be filter between the each other. It's called as to be censored glass filter. So this censored glass filter are present by David tube, means these two strain are not combined to be each other. Okay, just like it's a coli type first strain. Okay, and it's a second strain. So individually one strain are carried by followed by growth between the plates, so they are not followed. Okay, so these are combined to be together, means censored glass filter are removed. Okay, the censored glass filter are removed into David's tubes, then they are combined to be each other. Means S first strain are entered to be S2 and S2 strain are entered to be S first strain into the David's tube. Okay, just like okay. So they are combined to be each other, means completely followed by their ingredients, their factors also. So that timing this tube we are followed by together combined to be growth. So we are observed to be prototypes observed by growth. So easily indicated by bacterial strain. Okay, according to their growth we are considered to be their morphological characters also. Okay, and these are beneficial or harmful to be us also we are considered. Okay, just like a, so physical contact of the strain, for example, it's a F plus bacteria. Okay, carried to be physically contact, it's a F minus. So look at that, it's a F plus bacterial strain having to be fragile also. Means they are completely improved. They are having to be their genetic material. Okay. But F minus are do not having fragila, so they are not completely individual. Okay. So considered to be different between the each other. So that timing, these are called to be physical contact. This physical contact is called to be sex pilus. Okay. So two strain of the different strain, for example, male and female, they are contact to be physically contact between the each other with the help of the sex pillars, and that timing these are carried to be their genetic material are transformed to be this F minus cell. So they are also completely improved. Okay. So very important to be transform this genetic material between the each other, because they are made by new strain and new having to be inherited marker between the new strain. Okay, so consider to be different between the each other old and new strain also. Okay, so I hope guys you like to be this video, take by screenshot. It's a second procedure of the conjugation. So next procedure I am discuss with you what is the transduction process. Okay, because last video I am consider what is the genetic recombination of the transformation process and then conjugation process. Okay, so I hope you like to be this video, so share, like and subscribe my channel, and thank you.